It's a great pleasure for me today to introduce Herbert Tilg. Herbert Tilg and myself know each other already for a very long time because we are working on a very similar topic on liver inflammation, and therefore we from time to time saw us while we were still quite young and had more hair on our head and we're not gray hair, so this was a different time. But meanwhile, he has become head of the department in Halle, and he originally was at the University at Innsbruck between 1980 and 1986. He finished there with his MD thesis and then started into internal medicine in Innsbruck, and then went to a very prominent position in Boston to Dinarello lab. And he still is very close to Dinarello until now, because Dinarello has cloned several of the most important cytokines, and since then he has a very close relationship to him, and they still work together on different topics in this area. He then returned to Innsbruck and became there the vice head of the Department of Gastroenterology. And since 2002, he's heading the department in Halle of gastroenterology, which is a big department of 110 beds. And in parallel, he has still a lab at the university and is working quite hard, still in research. And he has two different topics. He has one topic about chronic bowel disease, but also he still is very strong in the liver. And today, therefore, we invited him in the SFB to give a talk about Adiponectin and liver diseases. Herbert, we are very happy to have you here, and we are waiting for your talk. Thank you very much for coming.
Okay, thank you, dear Christian, for inviting me. I'm coming from Tirol, so it's probably for you easier when I'm speaking English, because then you can probably understand me. So thank you very much for inviting. I think it's an interesting topic because we will speak about the interaction of adipose tissue, metabolism, and inflammation and liver inflammation, and I think that's a very timely topic. So I will focus on adipocytokines. I will speak a little bit about insulin resistance, and especially I want to give you my view that the adipose tissue is a major pro-inflammatory sink, and with respect to that might be of considerable relevance for liver diseases. Now, when we try to define adipocytokines, then we can have a very strict definition saying that those molecules, and those are the two prototypic adipocytokines, Adiponectin and leptin, that adipocytokines are only produced by adipocytes. But we know that in almost all obese patients, this tissue is infiltrated by macrophages, so we have different cell types in there. But by strict definition, adipocytokines are made by adipocytes. Now, as you can see here, for example, this is a staining for visfatin. I'll speak about this adipocytokine later. And this is not a liver tissue, this is gut tissue. The major question for us is, when you look at lean adipose tissue and then at obese adipose tissue, what is changing? And a lot of things are changing. For example, you can see the adipocytes become larger, they enlarge. It's a so-called adipose tissue hypertrophy. And you can see that many of those adipocytes undergo apoptosis. And we have infiltrating cells, infiltrating leukocytes. And it's not only mononuclear cells, macrophages, it's also T lymphocytes, it's also neutrophils, and it's also NK cells, for example. Meaning that in the adipose tissue, in the inflamed adipose tissue, we have many of those different cell types, and not only adipocytes, and they are producing various different mediators, not only, of course, adipocytokines, but also typical cytokines, and I will speak about that in the next minutes. So we have TNF-alpha, which is, and you are aware of that, a very potent pro-inflammatory cytokine. I'll speak about IL-6, which is also a pro-inflammatory cytokine, but which is also a very interesting cytokine for metabolism. And I will speak about adiponectin, which is a very potent anti-inflammatory adipocytokine, and which really nicely links metabolism with inflammation. This is in German, and Herbert Thaler, he has been an internist, a hepatologist, and he worked in Vienna, And actually, he described 45 years ago the diagnosis of fatty liver. And this is a very interesting paper because at the end, not the Mayo Clinic has described fatty liver disease or NASH. He described it because what he did liver biopsy in almost all patients. And he was also a pathologist. So he looked at all the histologies by himself. And that's the reason he made very careful observations. And for example, he described in that paper very clearly that fatty liver is something which you mainly observe in case of diabetes, or he called it at that time, of course it's metabolic syndrome, pre-diabetes. And what you also should be aware, and this is a very famous German internist, Wilhelm Epstein, and he described in this, and you should read this work, in the Berliner Klinischen Wochenschrift, 134 years ago, how he treated diabetes with salicylic acid, demonstrating for the first time that this is an inflammatory disease. So it's a very interesting paper to read, and you can get it. So the link inflammation and insulin resistance has been first, with respect to solid scientific data described in this science paper by Spiegelman 1993, and they have neutralized TNF-alpha in those FAFA rats, and they demonstrated that this causes a significant increase in the uptake of glucose in response to insulin. And also Hotomis-Ligel described in 1997 that in mice lacking TNF-alpha function, that they are protected from obesity-induced insulin resistance. This has been another important piece of evidence, this science paper, where they demonstrated reversal of obesity and diet-induced insulin resistance with salicylates, meaning that they scientifically did what Wilhelm Epstein did more than 100 years ago. And they really also demonstrated that targeted disruption of IKB kinase, which is part of the NF-kappa B pathway, is involved. And that at the end, insulin resistance, in many cases, is an inflammatory process. Now, TNF had been the first adipocytokine, and we can call it adipocytokine because it is also produced by adipocytes. Although, of course, monocytes, macrophages are the much more important producing cell type, but also adipocytes can produce it. So it has been, and this was actually described by Hotomis-Ligel beginning of the 90s, and at that time it was considered mainly as cachectin. So no one believed that TNF-alpha could be there in obesity. So this was really a concept which was unbelievable at that time. But it's very clear, it's elevated in most obese subjects, and we know that overexpression of TNF is at least partly responsible for insulin resistance in obese rodents and in humans. We also know from data in NASH that TNF and its receptor that the expression is high in patients with NASH, and the expression correlates with the degree of fibrosis, and expression is higher in patients with diabetes. And in experimental models, as you can see here, when you neutralize in the OB mouse TNF, as it has been done here with a specific antibody, then you can improve pathology. And interestingly, you can also improve steatosis, telling us that TNF, for example, is important also for the evolution of steatosis, not only for the evolution of inflammation. We know that we have, and this really tells us that NAFLD, non-alcoholic fatty liver disease, is a chronic inflammatory disease because we have now many data in hand with very different molecules also in the circulation, demonstrating that this is a chronic inflammatory disease. Elevated levels of TNF, of soluble TNF receptors, of IL-6, IL-1 alpha, CCL2, of course CRP and ferritin, reactive oxygen species, decreased levels of adiponectin, elevated levels of leptin. So we have really a lot of evidence telling us that metabolic disorders or metabolic inflammation is paralleled by these observations. The key question, of course, and we cannot answer this today, is how does, at the end, inflammation evolve in a fatty liver? And I'll give you now a little bit evidence that maybe the periphery, the adipose tissue, is very important. Now, this is a very exciting, important paper where they, first of all, made the observation that the high-fat diet causes activation of chunk. Chunk is a very important pathway in inflammation. And they show that it activates chunk in the adipose tissue. Now, what they have done is they have performed ablation of chunk-1 gene specifically in the adipose tissue. So those are adipose-specific chunk-1 knockout mice, and they demonstrated that by this way, you suppress high-fat diet-induced insulin resistance in the liver. And what they showed is that this effect is IL-6 mediated. And you have to keep in mind that what this paper tells us is that an adipose tissue-derived cytokine regulates a distal effect, namely an effect in the liver, which is hepatic insulin resistance. As you can see here, for example, the adipose tissue, and interestingly, this has been IL-6 specific. And you look here at mRNA expression in the adipose tissue, IL-6 goes down dramatically, TNF was not affected. And when you look at serum levels in those animals, again, IL-6 goes down, and TNF and other adipose cytokines, leptin and resistin, are not affected. So deletion of chunk-1 in the adipose tissue really impairs or reduces dramatically IL-6, and IL-6 is a mediator derived from the adipose tissue, which has distal effects in the liver, for example, upregulating insulin resistance via, as they have demonstrated in this paper, induction of SOX3. Now we have performed a clinical study in patients, in overweight patients who were undergoing bariatric surgery. And in those patients, we have taken biopsies, both from the liver and from the adipose tissue. And we have performed control biopsies after 6 months, so 6 months after surgery. And I will show you the data now. Here you can see IL-6 expression in subcutaneous fat versus liver. And you can see it's much higher in the adipose tissue than in the liver. And also when you compare visceral fat tissue biopsies, comparing with liver, again, it's much higher. And what you can see here, it's similar between subcutaneous and visceral fat tissue. And it has been very often claimed that the visceral fat is the major cytokine source, or IL-6 source in those patients. But as you can see here, the expression is similar. And when you look at DNF, for example, then you have again the same picture, meaning that in the adipose tissue, but also in the subcutaneous tissue, we have much more DNF compared to the liver. And those were all patients who were dramatically overweight, underwent bariatric surgery, and all patients then who lost weight. And when we now look at what happens after weight loss, which means after 6 months, and here are the results in the liver, then you can see the liver IL-6 expression goes down. But interestingly, the liver TNF expression did not change, but the liver SOX3 expression, and again, SOX3 is important for insulin resistance and is mainly under the control of IL-6. SOX3 expression goes down. And when you look now at the adipose tissue, and those are the data in the subcutaneous fat samples, then you can see that the IL-6 expression goes down dramatically, and also the DNF expression, but especially the IL-6 expression. So it's 26 times less after 6 months, which is quite impressive. And when you look at the anti-inflammatory adipocytokine adiponectin, you can see that the adiponectin expression again in the subcutaneous tissue goes up, but also expression of both receptors goes up, telling us that the cytokine cocktail in the adipose tissue changes from a pro-inflammatory tune towards, again, a more neutral or maybe an anti-inflammatory tune. Now, I stated that the visceral fat is important. And this is a paper published 2008 from Australia, where they looked at 38 patients and they performed MRI studies and did a liver biopsy. And they tried to show, and they also show it, that the visceral fat really correlates with the amount of inflammation in the liver. And as you can see here, for example, there was a significant difference between the amount of visceral fat and the inflammation in the liver. As you can see here, there was no difference with respect to steatosis, but again, as summarized here, when you look at the necroinflammatory grade or at the fibrosis stage, it was highly significant. The more visceral fat the patients had, the more inflammation they had in the liver, telling us that probably something is going on in the visceral fat, which at the end affects liver inflammation. And this again here is summarizing the data of this study. And when you do a multivariate analysis, it was again the visceral fat, but it was also IL-6 serum levels, which were highly correlated with the amount of inflammation, with the inflammatory grade, but also with the fibrosis stage, telling us that, of course, the visceral fat, but not only the visceral fat, also the subcutaneous fat are of importance and are an important cytokine source in those patients. So when we look at insulin resistance, and insulin resistance is observed in almost all patients with non-alcoholic fatty liver disease. It correlates with the amount of inflammation in the liver, and we believe today that in most cases insulin resistance is regulated via various inflammatory mediators. As you can see here, the insulin signaling, which is one of the most complex signaling pathways we have in biology, is affected via various pro-inflammatory stimuli like IL-6. I mentioned the SOC3 pathway, which interferes with insulin receptor substrate, but also IL-1 via AKK beta, or DNF via various mechanisms interferes with insulin receptor substrate and with insulin signaling, telling us that the pro-inflammatory cytokine cocktail, which we have in the adipose tissue, but of course also in the liver, interferes with insulin resistance, something we see and we observe commonly in patients with fatty liver disease. We know that cytokines in those patients, as stated, they control the inflammatory infiltrate in the liver, they regulate insulin resistance, and at the end, cytokines may mediate many aspects of chronic liver diseases, also in later stages, like, for example, fibrosis. Now, I will focus in the next slides a little bit more on typical adipocytokines, and I should say also that you can consider IL-6 also as an adipocytokine. Of course, it's a classical cytokine, but it's also an adipocytokine because we know that adipocytes are able to produce and to synthesize IL-6. Of course, and I stated this already, monocytes, macrophages can do this 10 times higher, but also adipocytes are able to synthesize IL-6. Now let's go to adiponectin. I've shown this slide already. Adiponectin is a very, very interesting mediator. It's synthesized mainly by adipocytes, but it's also expressed by various cell types in the body. And we don't know yet enough about it, but it's also expressed by muscle cells, by cardiac myocytes, and also by endothelial cells in the liver. It exists as a full-length protein or as a cleavage fragment, and you have to keep in mind that in health it circulates at very high concentrations, reflecting 0.05% of all plasma proteins. So it's very likely that it's very important in physiology or in health. As you can see here, it's mainly synthesized by adipocytes, and we believe today that the high molecular weight adiponectin is the powerful anti-inflammatory molecule, whereas the globular adiponectin or the low molecular weight adiponectin are not that powerful anti-inflammatory. It has been observed rather early by the Japanese, who also discovered adiponectin, that indeed low levels correlate very well with obesity, and I believe adiponectin is now the best studied adipocytokine or cytokine with respect to serum studies in the world. There are not hundreds, even more studies in humans on the role of adiponectin in various diseases. So the knowledge about the role of serum levels is very, very solid, and it's very clear. And also in other disease states which are associated with overweight, like non-alcoholic fatty liver disease, we know that levels decrease with advanced stages like steatosis or steatohepatitis. So it's very likely that adiponectin is also of interest and importance in human NAFLD. We know from adiponectin that it's a very powerful anti-steatotic molecule. And we have to keep in mind this because this is one of the key features of fatty liver disease. We have demonstrated that adiponectin and its receptors, as you can see here, are expressed in the liver, but the receptors are lower in NASH, as you can see here, and the adiponectin expression in the liver is, as expected, very low. Because we believe that the liver is a major target organ for adiponectin, meaning that it's mainly synthesized in the periphery, but the liver at the end is a major target organ for this important adipocytokine. We have also demonstrated that endothelial cells in the liver are at least able to transcribe adiponectin. We don't know yet whether the liver is really an important source of adiponectin. Probably not. It's probably more a target organ, as stated, but certainly we know it can be at least transcribed in the liver. And this is also supported by these data, and those are, again, our patients, overweight patients undergoing bariatric surgery. And when you look at serum levels, it's very expected that those go up after weight loss. This has been described repeatedly. But as you can see here, also the adiponectin expression, although low, goes up in the liver after weight reduction, and also the protein staining, as demonstrated here, goes up after weight reduction, meaning that at the end we have more adiponectin in the liver after weight loss. But there was no change in the expression of the receptors type 1 and type 2 after weight loss. So adiponectin is certainly in the hierarchy of all those molecules, of all those adipose tissue mediators, maybe the most important molecule, and it's really one of the big players in this cocktail which I have stated. And here I try to summarize that this molecule is a very, very pleiotropic cytokine. It suppresses pro-inflammatory mediators like TNF, IL-6, interferon gamma, and NF-kappa B. It has been demonstrated in various models of liver disease that it's protective, fatty liver, alcoholic liver disease, CCL4-induced liver fibrosis, other models of liver injury. Interestingly, it induces anti-inflammatory cytokines, for example, IL-10 or IL-1 receptor antagonist. It attenuates myocardial damage. It protects endothelial cells. It suppresses reactive oxygen species formation. It inhibits macrophage functions. And even it acts in the bone marrow, which is not a big surprise, but there it suppresses colony formation. So we have a molecule which is very important in health, which comes mainly from the periphery, from the adipose tissue, but which is able to affect many, many systems throughout the body. Leptin has been identified even before adiponectin, and we believe that leptin also, although it's not only synthesized by adipocytes, but it's mainly a pro-inflammatory. So it's really different from adiponectin, whereas adiponectin is mainly an anti-inflammatory. Adipocytokine leptin is more a pro-inflammatory. We know that high serum levels are observed in various autoimmune or immune-mediated diseases. And we also know that high serum levels are there in case of obesity, although the interaction of leptin with adiponectin is not well established and is not known. Now, going back to our obese patients after weight loss, you can see that serum levels go down, which is expected. But in the liver, there are no big differences between before weight loss and after weight loss, telling us that the leptin expression in the liver does not change, and we know that, for example, hepatocytes are able to synthesize leptin. But it will be, again, rather similar to adiponectin, meaning that the liver is mainly a target organ for leptin as it is for adiponectin, although, as you can see here, after weight loss, the two types of leptin receptors are decreasing, and we don't know exactly what this means. Resistin is a very strange adipocytokine because it's now almost known for 10 years, and we still don't know what it stands for or what it does, what it means. It's a 114 amino acid polypeptide. We know it exists in two forms, as a high and as a low molecular weight bioactive complex. Indeed, it's more a cytokine. It's not an adipocytokine because it's produced by almost all cells or many cells in the body, and it's mainly produced by monocytes and macrophages. But it's very clear it's pro-inflammatory, and this has been demonstrated in many models, but it does not really correlate with insulin resistance. So at the end, for the topic of insulin resistance, at least in humans, we believe it's not very important. And interestingly, it's one example where we see tremendous differences with respect to biology from mice to man. So that's really very obvious with resistin. And when you look again at our overweight population, you see that the resistin levels go up early, but then go down. The liver expression goes down, but the protein expression does not change. This brings me to the next molecule, which I think is very interesting and first of all, it has a very interesting history. It's PBEF. PBEF stands for pre-B-cell enhancing factor, NAMT, or visfatin. Now, what about the history? Shimomura's group, they had identified a novel adipocytokine in a science paper and called it visfatin. And they claimed in this science paper, which was published, I guess, 2007, that visfatin binds to the insulin receptor. And this made it interesting at that time. But on the other side, then it had been realized at that time that the sequence of the gene product corresponds to a molecule which had been described, namely as pre-B-cell enhancing factor, PBEF. And PBEF enhances the effect of IL-7 and stem cell factor on a pre-B-cell colony formation, telling us it's a cytokine which is important for the B-cell development. And PBEF had been demonstrated being involved in inflammatory conditions and also that it exerts anti-apoptotic properties. Again, a few years later, but before it had been described as visfatin, the same molecule was identified as the nicotinamide phosphoribosyl transferase. So the same molecule, PBEF, is identical to an enzyme which has an intracellular function. I will speak about that, which is critical for the synthesis of NAD. And you know NAD is important for everything because it's essential for the energy metabolism of the cell. So we have a molecule which has three names, but it has been demonstrated, or it evolved from three different areas, but it's the same molecule. And unfortunately, and I think this paper had been retracted because no one could demonstrate or repeat that it really binds to the insulin receptor. But still, this molecule is very exciting, but it helped it to bring it to the scientific community, so we could have started to investigate what it stands for. Now, we have published 3 years ago in the Journal of Immunology that recombinant human PBEF or NAMT is a pro-inflammatory cytokine. So when you, for example, stimulate monocytes or macrophages with recombinant human PBEF, you can induce a lot of pro-inflammatory but also anti-inflammatory cytokines, and I won't go into details with all those data, but we have shown, for example, in inflammatory bowel disease that PBEF is highly expressed in the gut tissue, correlates very nicely with other inflammatory parameters. So it's really a pro-inflammatory cytokine, and when you look again at our obese patients, you can see that the serum levels after weight loss go down, and also the mRNA expression in the liver goes down. So this fits very well with the findings we had that it's mainly a pro-inflammatory cytokine. Here you can see liver tissue expression of numbed BBEF with statin before and after weight loss, and again its liver expression goes down. Now, here is a graph which summarizes the biological functions of BBEF numbed, and of course everything is still in a rather early phase, and we don't know a lot about it yet, but I want to focus on a few aspects. First of all, it's extracellular, active as a cytokine. And this is mainly pro-inflammatory, and we have, for example, shown that it activates chunk B38, BI3 kinase, so it's really a pro-inflammatory cytokine. But its main function is intracellularly as an enzyme. And as an enzyme, it's the key, the rate-limiting step of the formation of NAD out of NAM, nicotinamide. So it's a phosphoribosyl transferase, and via this activity, at the end, we have NAD, which is essential for many, many functions in the cell. So it's really, it has these diverse properties, those diverse functions. And in the last 1-2 years, several other interesting aspects have been demonstrated. For example, that it's probably really involved via SIRT1. SIRT1 is a histone deacetylase. Those molecules are very important also for metabolic aspects. And via SIRT1, for example, it regulates circadian rhythms. We also know that NAD activates DNF. And we know that this has been last year a Nature Medicine paper where they showed that this loop works via SIRT6. So sirtuins are obviously not only very exciting, interesting molecules in the last 1-2 years, but they are also involved in those pathways. And it's obvious that those are aspects which are certainly associated with inflammatory pathways. We know there is a specific inhibitor of this enzyme, and I will speak about that in the next slides. We were now interested in Con-A hepatitis. And to find out the role of NAMT with fatine in this model. And you can see here at the mRNA level there is a dramatic increase of its expression in those animals after stimulation with Con-A. And we tried, and I'll show you the data, to find out when you block NAMT via this specific inhibitor what happens, and also what happens when you overexpress BBEF NAMT via hydrodynamic perfusion, and also some in vitro analysis. Now, FKA66 is this specific inhibitor, and this inhibitor had been developed as an agent to induce apoptosis. And this FK866 is used at the moment in phase 2-3 studies in various cancers. But it's a very specific inhibitor of NAMT, and as you can see here in those histology data, when you give it to the animals at the same time when you give Con-A, you can see a dramatic decrease in inflammation in the liver, and this is summarized here. You can see here the transaminases, so those are the animals receiving FK866. And when you look at various cytokines, CXCL1, IL-6, IL-1 beta, interferon gamma, TNF-alpha, you can see that it really very potently suppresses inflammation. When you do some in vitro studies, for example, with hepatocyte cell line FL83B, you can also see that when you use this inhibitor again, you can in vitro suppress the production of CXCKC. So it's really anti-inflammatory also in vitro, and those are the results of experiments where Alexander Moschen did a hydrodynamic perfusion experiment. And as you can see here, when you use the BBEF, then you can induce more inflammation, as demonstrated here. Those animals have higher transaminases, and also with respect to various cytokines, he could demonstrate that CXCL1, IL-6, or IL-1 beta are higher expressed in those animals. So telling us when you either block it via NAMT or when you regulate it up via this technique, you really have a nice picture of its biologic function, at least in this animal model. And when you neutralize it in vitro, as demonstrated here, using silencing experiments, you can see that when you silence NAMT with fatin in vitro and you stimulate cells, for example, with DNF or LTA or with LPS, you can again suppress CXCL1 or CXCKC, telling us that also in vitro suppression of NAMT, of this enzyme in this case, and in this case via the silencing technique, is anti-inflammatory. So visfatin is an adipocytokine, but it's more a cytokine again. And one of my messages for you is that we cannot easily separate those aspects because many cytokines are adipocytokines and many adipocytokines are cytokines. But we have to recognize that those mediators, and this is true, for example, for visfatin or NAMT, are produced by many cells throughout the body. But it became very clear in the last years that non-alcoholic fatty liver disease is a chronic inflammatory disease where we certainly have this imbalance in the cytokine cocktail. And of course, we don't know exactly how the processes in the adipose tissue evolve, what is first. But it's definitely the case that we have many more pro-inflammatory mediators around than anti-inflammatory. Certainly, the whole process starts with lipid accumulation, which is the first step. But as discussed in my presentation, the other hits are very unclear. And of course, it's not only endotoxin probably from the gut. It's many other hits which are attacking the liver. And we have to keep in mind, and this is one of my messages here, and I think it's supported both by animal experiments but also by our human data, that adipocytokines from the adipose tissue or pro-inflammatory cytokines from the adipose tissue are of key importance for the phenotype of this disease and for the disease process in the liver. So, thank you very much.